All right. Welcome to Gen Bio 2, continuing along with plants and plant diversity. We have the beginnings of the seed plants and a rogue gecko. Seed plants first appear in the fossil record about 305 million years ago, and a lumbar thing caused a gecko to disappear. Come here, you little wanker. Everything's fine. There was a accidental weapons discharge, but everything's fine here. How are things on your floor? Okay, so 305 million years ago, we have the origin of extant seed plants, right? So roughly in the fossil record, 360 million years ago is when seed plants first appear. So the seed plants consist of two groups: the gymnosperms and the angiosperms. Good times. Angiosperms by far being the most successful of all terrestrial plants. So, quick review of the plant groups and some basics about them. The mosses and the other non-vascular plants were gametophyte dominant. Their sporophyte was reduced and dependent on the parent plant for nutrition. So all the green stuff was haploid gametophyte, and the diploid sporophyte was this highly reduced structure. So then we get into our seedless vascular plants, including ferns, and we had our gametophyte being reduced. It's small, but it's also independent. Both the sporophyte and the gametophyte stages are independent. For most of the seedless vascular plants, that means that the gametophyte is photosynthetic and thus able to get nutrients on its own and lives free on its own. Then we have the sporophyte over here, which is the dominant life stage. And also the sporophyte is where we find our vascular tissue, right? Being roots, leaves, stems, that sort of stuff. So none of that is present in gametophytes. All right, and now for the group that we are going to be talking about today, the seed plants, the gymnosperms and the angiosperms. So the gametophyte is generally microscopic at this point, and now it is completely dependent on the sporophyte for nutrition. So kind of hit the reverse of our mosses and non-vascular plants there. That's kind of fun. And we'll get into the big features of each group. So, both seeds and pollen grains, key adaptations for life on land. So we've got a gametophyte that has been reduced to a mostly microscopic stage. Basically, spores remain within the sporangia. So microscopic gametophytes develop from, so we have the sporangia. Let's just say this is the sporangia. And it makes, so diploid sporangia. And here's some spore mother cells. And then they do your good old old-fashioned meiosis, and we get spores. And then the spores germinate, and we get, our gametophyte. All right here, contained within the parent plant, often within the sporangium itself. So, this is great. The gametophyte is now completely protected against environmental stressors. So, just checking, he's still there. So, it protects against environmental stressors. So, UV damage, and drying out. Who remembers what that's called? The fancy science word for drying out and having a bad time? Is it desiccation? I think it is. So there you go. And then this gametophyte is completely dependent on the parent plant. So, not only is the parent plant providing it protection against environmental stressors, it is also providing it all its nutrients. So, hooray! So, over here we have this beautiful diagram of a seed right here. The integument is the outer wall of the seed. So, then lining the inside of the integument, we have the sporangium. Since this is the seed, right, it's going to produce the egg. So it's going to be the female gametophyte. Female gametophyte, big gamete. So, if it's related to an egg, you tack mega onto anything that will be associated with that. So, this sporangium is the megasporangium, which produces the megaspore, right? And then the megaspore produces the gametophyte, right here in green, which I just filled in with purple. And then the gametophyte produces the egg. And then these megaspores over here are all failed megaspore tissue. They all dead. They bad. So sad. Okay, so not too terrible. Okay, so let's go into heterospory. Hetero, different spores, kinds of spores. So, separate spores for producing sperm. So, the gametophyte that produces sperm, and the gametophyte that produces eggs. And what's that? Well, let's have a microspore. And it produces the microgametophyte that produces sperm. And then let's have a megaspore that produces the megagametophyte that produces eggs. So, megasporangia for making megaspores that produce megagametophytes that produce eggs. Big deal about this. is basically you have your megaspore mother cell. Change the ink color. Red's a classic. Megaspore mother cell. Mother cell just means it's going to do the type of mitotic, the cell division that gets you the thing it's named for. So a mother cell for megaspores is going to do meiosis and get you a megaspore. A megagametophyte, an egg mother cell is going to do mitosis and get you an egg. Because it's plants and gametes are made by mitosis. Okay. So here's the neat thing about plants. You have the megasporangium that produces the megaspore mother cell that undergoes good old-fashioned meiosis. And we get the product of meiosis, 4 spores, but 3 of them are aborted and do not produce an egg. Only 1 spore survives. 4 spores enter, 1 spore survives. It's the Thunderdome of Megasporangia, the Megasporangia Thunderdome. There is no Thunderdome for Microsporangia. That microspore mother cell, you'd think it would be a father cell, but nope, still mother cell. The microspore mother cell that makes sperms, all 4 spores crank out sperm. So, hooray. There you are. Okay. You're still with me. All right. Not too bad. So, microspore develops into the pollen grain, which is pretty cool. So, we'll talk about that in a hot second. So, not so bad so far. This is 2 different kinds of pine cones, which we will talk about in gymnosperms. One is an ovulate cone, and the other is a staminate cone. Ovule, stamen, so, not too terrible. All right, hopefully. Anyway, ovule is the one to remember. Ovule, egg. The ovule makes the egg, so the ovulate cone is the one that has the eggs that will then be, fertilized to get us an embryo that's protected by a seed. Whereas the staminate cone is not the ovulate cone, so it cranks out the sperm, the pollen. Okay. So, talking about pollen. Basically, you have the microspore. So, here's my microspore. Then I have my microgametophyte. Yeah, that fit on there. That looks good. And now I produce my spoime. So, microspore mother cell, undergoes meiosis, and I get my sperm nucleus. So, not too terrible. So it's all within there. The microgametophyte and the microspore make up the pollen grain wall. So I may have been slightly discombobulated when I talked about this in person. If that happened, ignore that guy. Anyway, not too terrible. The microgametophyte is within the microspore. Microgametophyte is kind of a fancy way of saying male gametophyte, just a reminder. So, interchangeable. Anyway, and then the gametophyte cranks out the actual sperm nuclei. All right, this means that we are no longer dependent on water for fertilization. So there are two basic ways we can spread pollen now. Wind. Whoop, that's not looking good. Wind and critters. Not this kind of critter. This kind of critter is being unusually active. Let me see if I can calm him down. Sometimes if I just throw him under my hair, he goes to bed. Okay. So, mostly wind and critter-based pollination, right? So this is our way of fertilizing. Before seed plants, fertilization was dependent on water. Also, before seed plants, sperm had flagellum. There are a couple of gymnosperms that have flagellated sperm, indicating just how primitive they actually are, which is kind of cool. Okay, so boop. This is a pretty cool video, a little TED Talk, in which the guy goes through electron micrograph images of pollen and talks about the different methods those pollen have for pollination and getting around. And no, we're not going to watch a video within a video. That's on you. This is the website here. And it's also in the student lectures, so you can open that up and copy and paste that right in. Highly recommend it. It's actually a very cool TED Talk. So, totally should do it. All right. Not so awful. All right. Seeds and pollen grains are key adaptations for life online. Let's see. Let's hit my seed slide. There we go. Okay. So ovules and production of eggs. Now that we talked about the micros, let's talk about the megas. Okay, so first off, the integument. Think of it like the future seed wall. Right? And it is sporophyte tissue. And without getting discombobulated, it is not sporangium. So it's literally just a thick layer of tissue that will become the protective seed wall. Not too terrible. So it protects the megasporangium. Okay, so we are going to have integument. I know there's literally a picture there. This tiny little opening is basically how we get sperm to egg. So we'll talk about that in more detail a little later when we talk about actual pollination in seed plants. But that's a little opening for sperm to egg. It will fuse after fertilization. Okay, then we have a very thin layer of, well, okay, so we have our megasporangium. That looks good. Then we have our megaspore. Let's go green because it's green plants. Remember, only one megaspore survives. The green and the blue don't contrast really great. Then that megaspore produces this itty bitty multicellular gametophyte. Just itty bitty multicellular gametophyte. Whoa, running out of space, man. Okay. Hold. Or I could erase everything. While I'm trying to fill that in. I'm so gonna cut this for time. Well, not cut the entire thing. I'm gonna cut this drawing time down. Here I am narrating stuff once. Okay. That's called good enough. There's our mega gametophyte. And then our final layer of our nesting doll is an egg. Woo! So that's pretty neat. That megaspore is kind of cool. So we have our haploid megaspore. So over here, hopefully you drew with me instead of just staring at this, admittedly nicer in graphic, but honestly, multiple studies show that just staring at a figure provokes very little memory retention. Drawing gets you memory retention. So that's why I draw, so you draw with me. Anyway, so we have the integument layer. We have the megasporangium. we have the megaspore, which we have a little teeny spore wall, still 1 cell thick. Then we have our megaspore internals. And then we have the megagametophyte with the egg. And that megagametophyte is highly reduced. Highly reduced. Okay. So, an ovule is a term for not yet fertilized. So, basically, the ovule is the whole structure that contains the unfertilized egg. It will go from fertilized ovule to seed very quickly. So when I throw around ovule, think before seed, right? Like, which shorthand before fertilization. If you want to get super technical, the integument has to fuse, and you have to get the development of an embryo, and you have to get like the separation of the integument from the layer holding it to get like, and the integument has to harden to a seed, blah, 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 blah. And until then, it's sort of like an ovule transitioning to seed. But that would be like a super dick move to test you over. So that's like technical information for you, that ovules, is a term that technically includes that transitional stage. But when I use it, I'm generally referring to before fertilization, the egg-containing structure. And it basically shakes out to megasporangium, megaspore, and integument. So let's write it out to end. So integument, megasporangium, megaspore, and then megagametophyte and egg. So, whee! Pretty cool. Yes, things have gotten a lot more complicated, but at the same time, it's kind of all still the same thing. That basic life cycle holds. All the vocab for the basic life cycle hold. If I wanted to, we could go, microscopic enough to the cells, and I could point out what makes up the archegonium in this highly reduced gametophyte. Not that that's really worth doing, but there is still antheridium and archegonium even at this super highly reduced stage. That pollen grain, there's still antheridium in that microgametophyte that produces our sperm nuclei. So, yeah, all the vocab holds. It's just gotten, Russian nesting dolled on the sporophyte. Okay. Boop. Okay, let's talk about the advantage of seeds and pollen grains. Right? So basically seeds. These are our new distribution mechanism. Right? In mosses and the seedless vascular plants, right? So in the non-vascular plants and the seedless vascular plants, the mechanism for distribution was spores, right? So non-vascular and seedless vascular plants, spores distribute the plants. What's the advantage here? Seeds over spores. 1, highly well-developed protection compared to a spore. A spore wall is always one cell thick. Even on our, highly reduced, plants here that have sporangium retained inside the sporophyte and all that stuff, the spore wall is one cell thick. So a wall itself helps protect against desiccation, but the plants that require spores for distribution, well, they're kind of restricted to extremely humid areas with a lot of standing or flowing water. This is because one cell thick of protection is some protection, right? But it ain't great. Right? It's like, something is better than nothing. But, the integument that becomes the seed wall, multiple layers thick. Right? So the integument the sporangium and the megaspore sort of all fuse together to get our seed wall. Granted, when you get down to it, the integument is really doing the heavy lifting on that one. It's almost all integument, but still, what's left of the megasporangium and the megaspore also make up that extremely thick cell wall. So what you get is a very hard, thick seed coat. This is highly resistant to environmental conditions. Right? Not only extreme desiccating conditions, but seeds are pretty resistant to being flooded. Right? You submerge terrestrial plant spores and they're not going to have a good time. You submerge seeds and they can float around until they get washed up somewhere where it's better. Right? And that goes hand in hand, being able to be highly resistant to poor environmental conditions. Let's write it out. Poor, let's change colors too. Highly resistant to poor environmental conditions. Goes hand in hand with the next two bits. Dormancy and food. So one, let's kind of reverse these real quick. Embryos have a built-in food supply. So there is food tissue within the seed. This can keep an embryo functional for quite a while, especially when they go dormant. Dormant means they could go into extremely low metabolic rates. Yeah, it just had to be over the red fruit part of that seed. Extremely low metabolic rates, at which point their nutrient needs and water needs are almost nil. Like, this is true dormancy. Almost zero requirement for nutrients and water. Some of these seeds can go into dormancy for ludicrous amounts of time. Ludicrous amounts of time. Right? There are seeds in Yellowstone that have been dropped from pine cones, actually, there are seeds in Yellowstone that have been dormant in their pine cone, and they basically remain dormant decades, even centuries, until there's a fire hot enough to cause those pine cones to pop. So the pine cones basically don't open unless there's enough fire that a lot of the little plants in the undergrowth that would compete with the seeds germinating have been cleared out. So that's why some of those cool pine trees in Yellowstone only pop under high heat, because high heat means big fire. Big fire means all the underbrush got cleared out by big fire. And so now when you pop and drop those seeds, they're going to have great conditions for growth. So decades, centuries of dormancy there, but you want real insane kind of dormancies. 65-ish million years ago, there was a big kind of FU asteroid that hit off the Yucatan Peninsula, the Chicxulub crater, and kind of kicked off a chain reaction of events that led to the extinction of all the dinosaurs. And part of that chain reaction of events was enough stuff thrown into the air from the meteor impact and from the volcanoes around that there were, thousands of years of almost no sunlight. And basically most plant life died out and was carried on through dormant seeds. That's some serious dormancy right there. So that's kind of cool. All right. So these seeds are cool. Very cool. That very thick seed coat that protects them from environmental conditions, that inborn food tissue that helps supply them with nutrients, the ability to go dormant where they have extremely low to almost no metabolic rates while waiting for ideal conditions, right? Between dormancy and food, you can wait to germinate. Hey, that rhymes. Wait to germinate until conditions are good. Until chef's kiss. Mwah. Conditions. Right? Not enough light, not enough water. Stay in that seed. All right. Other things that are cool. Seeds allow for enhanced dispersal mechanisms. There's a lot going on. Seeds, a lot of seeds, right, can survive gastrointestinal tracts. So what that means is herbivores and frugivores, fruit eaters, distribute the seeds, which is great because they have an ability that most plants don't, and that's motility. They can move around, baby. So something eats them seeds, wanders around, and then drops the seeds. And good news, it gets dropped out in poop, which is fertilizer. So that's great. They can survive the GI tract. Coconuts float. Right? So there's my coconut. They have an extremely thick, hard seed coat, and they can float on the ocean and travel vast distances before being washed up into places where conditions are good for germination. So that's pretty cool. And then E is literally kind of right back up to what I said in A, because I went beyond good protection and talked about what they protect from. And then apparently I had E where I listed what they're protected from. So that was solid slide design by me. Mwah. Good job, Past John. Okay, now we're on to gymnosperms. Woo, all that, halfway in, and it's gymnosperm time. Okay, so features of gymnosperms. Woody tissue is extremely common amongst the gymnosperms. I wouldn't say it's diagnostic of the gymnosperms, but woody tissue is a key adaptation that we first see in gymnosperms. Ferns do not have a woody tissue. They do not have, technically speaking, what we call wood. Gymnosperms are the first plants where we see wood. All right. So, incomplete ovule. What does that mean? The ovule is exposed to the outside. Let's talk about why that is, and why that is has to do with strobili, which are cones. So cones here in gymnosperms are where the ovules are basically where we produce our gametes. Right, so there are male cones and female cones. And so what happens is these ovules develop on scales. So that is a scale. 2 ovules to a scale. So when you look at the pine cone, we have the scales. A growing female pine cone is not a popped open one. This is a pine cone that has been sort of artificially opened up. The ovules are not fully exposed like this. This is actually probably fruits of some sort. I don't know. Not fruits. These are gymnosperms. Excuse my terminology. Fruit is technically an angiosperm thing. So these are probably more seed than ovule. Like, I don't know. I think someone just artificially opened that sucker up so that you could see the ovules. That is way too fresh for a pine cone to be that open. So anyway, that's just so you can see the ovules. So we've got the scale. From the side view, in purple, we have the scale. All right. Then we have the ovule sitting on the scale. And then if you look at any of these, we have blank space. And that blank space, no matter how thin it gets over here, is open to the outside. Not super duper open to the outside, not at all naturally occurring in the way that this picture is. But this right here, this cross-section of a pine cone is much more indicative of the kind of open to the outside that we see in our ovules. So we can see continuous blank space all the way down to the ovule. So not super duper open to the outside, but open to the outside. This is important because pollen have to reach the ovule, right? Pollen must reach the ovule. And it can't do that from the tip of the scale. So if I get a fanciful color, let's go purple. No, a lot of that electron microscopic electrons are purple, orange. Orange is usually pretty bright. So we've got our little pollen grain, not to scale. This is too big. And it goes whee, and then lands back here. So, and that has to be wind distributed too. So, not super open to the outside, but open to the outside because pollen must reach the ovule. So, there we go. Not too bad. This other one here, you can define gymnosperms by what they don't have compared to angiosperms. No flowers or fruit. So, not like the greatest thing, but when we're just comparing gymnosperms and angiosperms, they don't have flowers or fruits. And you'll never guess when we get to angiosperms what one of the big defining things of those is. Woo! But I'm getting ahead of myself. Spoilers! Anyway, okay, so gymnosperm literally means naked seed. Hence, the ovule is exposed to the outside. It's naked. It's not completely enclosed. All right. So, here's another good image of an ovule on the scale. So, here's our scale. And then just to get an idea of just how small of a section or small of a structure we're dealing with, megaspore mother cell, pointing to this tiny little thing here. But still, outside. And woo, ovule. All right. You can compare that to angiosperms here. There is no ovule facing outside. Right? So, ovule is completely enclosed. And if it's completely enclosed by tissue, an ovule or multiple ovules completely enclosed by a structure means you have an ovary. An ovary completely surrounds and encloses one or more ovules. Good times. Okay, let's go into the actual diversity of these things. You'll see this, like you can ignore this for now. We'll see that in much more detail later on in angiosperms. Okay. Diversity. We are going to talk about Coniferophyta, Cycadophyta, Gnetophyta, and Ginkgophyta. If you've been in the Bio 2 lab, you've seen these. Gecko is still here? Yes, he went to sleep in my hair. That's what I thought. Okay, so diversity. Going to be pretty cool. This is a cycad. This here is a cone. Pretty cool. All right. So, I'll go through this stuff as we go. So first, let's talk about reproduction. All right, heterosporous, because it's always heterosporous from here on out. Right? Megaspore, microspore. So, microsporocyte. A sporocyte makes a spore. Same thing as mother cell. Okay. So, the microsporocytes undergo, let's go back to red because I feel like it has the best contrast, meiosis. And they produce microspores. And the microspores produce the pollen grain. The inside lining of which being a gametophyte, which then cranks out our sperm nucleus. Wow, that almost spelled nucleus, whatever. And just a little reminder, it probably makes a lot more sense here. So red is the microspore. So the microsporocyte, the microspore mother cell, produces the microspore. The microspore germinates a thin layer of gametophyte inside. So this is the pollen grain. And the pollen grain contains the sperm nucleus. Right, a lot better as a drawing than it is a little line diagram. Anyway, megasporocyte. Again, who could have guessed? Meiosis. And then we get megaspores. By the way, in case your brain was still stuck on, so I have had people ask in the past why I draw 2 sperm nuclei and not 4. Right? The reason for that is this is plants. Right? Meiosis gets us the spore. So meiosis gets us 4 pollen grains. We crank out a gamete from mitosis, which just gets you 2 cells. So 4 pollen grains, each containing 2 sperm nuclei, only one of which is actually functional in gymnosperms. So, a quick throwback. All right, so meiosis gets us our megaspores and 3 crash and burn. 3, let's just say die. So we get one surviving megaspore. That surviving megaspore is going to do its meiosis, right? Oh, sorry. That surviving megaspore does mitosis. See, it even still gets me. I'm even just reminding you about it. We do our mitosis and we get our gametophyte, our mega gametophyte tissue. And we get our egg. And basically, this whole stuff here is an ovule, but better as a graphic. Okay, so kind of cool. Both of these are going to be produced inside cones. This is an ovulate cone. Again, when you hear pine cone, think ovulate cone. The staminate cones are that weird papery cone that looks all weird. So here, generally at the very tips of branches, which makes sense. Okay, so let's ask two questions. One, I said it makes sense. Why are staminate cones at the tips of branches? So, why are our pollen producing structures at the tips of our branches? Shout out, yell, make the people around you confused as to why you're talking to the computer screen. Look bashful and say you're playing an online game. I don't know. Wind, right? Our distribution mechanism for pollen is wind or critters. And in gymnosperms, it's not critters. Gymnosperm pollen is wind distributed. So pollen is wind distributed only. So, staminate cones are at the end of branches. Pollen cones are at the tip of the branches because that's where they'll catch the most wind. So that's our first question. The second question, ovulate cones tend to be higher in the tree than staminate cones. So, boop, 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 boop. Nice drawing. Okay. So here's some staminate cones. And then here's some ovulate cones. This is looking unnecessarily Yuletide, but whatever. Why are ovulate cones higher up in the tree? Shout. Shout. Let it all out. These are the things. I lost it. Come on. Anyway, what distributes pollen? Wind. And when that wind comes a blowing by, woo! and starts knocking that pollen out into the wind, it's probably not very likely that it's going to go up and to its own combs. This is prevention of self-fertilization. Prevents self-fertilization. Okay, other cool thing, right? So, we've got our ovule. Sitting on our scale. And let's just do another scale right here. Right? Okay. And we'll draw an ovule there, but not care too much about it. All right. So, I'm not going to go through all of the tissues once more, but we'll do like a lazy Russian nesting doll of tissues. Egg. All right. So, I got my pollen grain. And it's going wee woo, wee woo, wee woo on the wind, and it eventually makes it to the scale and eventually lands here at the opening to the ovule, the little gap in the integument. And we've got a minor problem, and that's a bunch of nesting doll of tissues before we can reach the egg. And what do we do about that? Well, we grow a tube. The, 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 the I've even lost track of how many layers I have here. I don't know. I'll just do one more. There's my egg. Pollen grain. My pollen grain has, despite all odds, managed to land over at the ovule. And we are going to have a pollen tube grow. And it basically is literally just a little invasive tube that grows through the tissue layers and connects up to where the egg is. And once that pollen tube grows through, and it is very slow growing in gymnosperms, right? we have a slow growing pollen tube. In some of those very slow growing trees, this very short distance can take a year for that pollen tube to cross. So that's kind of nutty. But finally, once that pollen tube connects the grain to the egg, the sperm are released into the tube. Sperm nucleus, only once, only one. Remember, I keep saying that in gymnosperms, only one sperm is a functional nucleus. The sperm nucleus, singular, this is actually going to be important when we get into angiosperms, hence why I'm driving it home. Sperm nucleus travels through the tube and ferts the egg. Woo, I barely made enough space. Okay. So, this is, voila, the pollen tube. Like I said, wind has to be in very good conditions. Like, you have to get really nice wind conditions to get that teeny tiny, and I am really underselling how small pollen grains are. You know, if they were the scale at which I drew it here, which is great for actually making a drawing, but if they were at this scale, it would be impossible. Like, one pollen grain would barely fit into that tiny gap between scales in that ovulate cone. No. What happens here is that pollen grains are tiny to a gobsmackingly amazing degree. And so when you get a bunch of wind and those pollen grains are passing by an ovulate cone, it's actually, a numbers game on how many of those pollen grains can make it all the way across that scale and bump into the ovule there. So, vast amounts of pollen are released. Vast amounts of pollen. So much that even though these grains are microscopic, you can see it on your car. Think of just how much pollen has to be released when you park under a pine tree and see your car dusted yellow. Those grains are microscopic. The amount of grains required to see yellow dust is hard to mentally grasp. So, literally, why we can have this happen when it's literally wind blowing through a tiny gap in a scale to make it all the way back to the back end of the scale where the ovule opening actually is. But it works out. Works out enough that gymnosperms are, a lot more common than the seedless vascular plants and a lot more common anyway. So, gymnosperms are doing all right. Okay. Turn that off. Oh, this is just a beautiful slide. Anyway, so the pollen tube is what allows us to connect. So it basically, the tip of the tube releases digestive enzymes and literally digests gametophyte tissues. It digests all those tissue layers until it actually reaches the archegonium. Yeah. Like a year to grow the pollen tube. Some species even longer. Like the longest reproductive cycle in some of these gymnosperms is 3 years from pollen landing at the integument to functional seed. Whew. Okay, so here is your large, more inclusive life cycle, including this stuff. Again, it's going to be very similar to the original life cycle, right? We still have, sporophyte. We still have sporangia. Right? And we still produce spores. And those spores still produce gametophytes, which have the requisite tissues on them. And you still get fertilization, the sperm meeting the egg, and you still get the zygote and then the new sporophyte embryo. So, here are food reserves, worth noting, are derived from the gametophyte, right? And so they are haploid food. I'm emphasizing this in gymnosperms because it's different in angiosperms. So this is going to be one of our differences here. So we also have a single seed coat. Another one of those previews of coming events for differences between gymnosperms and angiosperms. And remember how I said singular nucleus as a okay, so we've still got all our basic vocab. We just have to add in a few things. One, heterosporous, so micro and mega. Two, remember mother cell and sporocyte, are like the same diff. So the megaspore mother cell produces the megaspore. So there you go. This life cycle drawing, kind of annoying. I like it better when, just in my head. Vertical means, going through the sequential changes. Here, they kind of just fit things in where it fit. It's from the book. It's pretty. And hint, hint, it'll be printed on an exam page. But anyway, you have your ovulate cone, right? That's some vocab we have to add in there. And the ovulate cone has the megasporangium, which produces the megasporocyte, which undergoes meiosis to get the 4 megaspores, only one of which survives. The megaspore germinates and produces the gametophyte. Oops, I don't want to put that there. So the gametophyte contains the archegonium, because mega, egg, meg, meg, yeah, I just thought of that. I'm a genius. Anyway. Then you have the pollen grain landing. It makes a pollen tube, and the sperm nucleus travels through the pollen tube to the egg nucleus. Fertilization, zygote. We get growth of our zygote within our ovule. It becomes an embryo. And as it becomes an embryo, that integument fuses up, and we have a genuine seed. So, there we go. The seed having its gametophyte-derived food reserves that are haploid and are single seed coat. The seed eventually, in gymnosperms, generally carried on the wind. A lot of these seeds have fun little adaptations there. A lot of pine cones drop helicopter seeds, so that's kind of fun. The ones that spin slowly. I like those ones. Anyway, and then seed germinates, seedling, sporophyte. So there we go. And then over here, on the other hand, just continuing the sporophyte life cycle. We're heterosporous. So we have the pollen cone, the staminate cone. And it has the microsporangia, which produces the microsporocytes, which undergo meiosis to get the spores that produce the gametophyte that produces 2 sperm nuclei, of which only 1 is functional. So, complicated with new vocab, but fundamentally the same alternation of generations. You just got to add the new vocab in and kind of visualize that stuff. So, haploid, gametophyte stuff here, sporophyte stuff all over here. Still alternation of generations. Okay, that's gymnosperms. So now let's get into angiosperms. Angiosperms are the newest and most successful plants on the block. Now, originally thought, whoa, that is a poorly spelled thought. Originally thought to have evolved 140 million years ago. There is new evidence from 2013, so not even really new, 11-year-old evidence. So we've known for more than 10 years that they are much older than that. 240 million years ago. So we found fossilized pollen grains. The pollen grains that had the critter vector. Pollen adapted for critters. That is, really, you have to kind of just believe me that spells critters. So one of the big deals about angiosperms is that angiosperms co-evolved with pollinators. Mostly insects, but not always, hence critters. In other words, here, critters carry the pollen. So, not all angiosperms today require critters to pollinate, but the origin of angiosperms is the origin of pollinators. Not too terrible. So, that pollen grain that they found in the Triassic era, dated to 240 million years ago, the early Triassic, man, it had tiny little adaptations for sticking to bugs, probably beetles, because bees did not exist 240 million years ago that we're aware of. All right. If you make a clade of the seed plants, it is a genuine clade. They share common ancestry with the gymnosperms, so you can make a clade out of seed plants. Remember, a clade is organisms all like a single common ancestor and all the descendants related to it. So common ancestry with gymnosperms. Seed plants equals a genuine clade. Probably has a fancier name than seed plants. All right. So, synapomorphies, shared derived characters that separate angiosperms from their ancestors. The key developments that produce the angiosperms. 1. An ovary, which you can think of as fully enclosed ovule. Sometimes plural. A lot of the times plural. Right? That's a big synapomorphy. All right. Another. Pollination, rather than occurring at the ovule, occurs at the stigma, because the pollen grain can't reach the ovule, because the ovule is fully enclosed by an ovary. So, gymnosperms, ovule open to the outside, the pollen grain actually floats in and connects up with the ovule. Here, the pollen hits the stigma. Yeah, red flower. So this is about to get the stigma is the site of fertilization. So when the pollen lands, it lands on the stigma. When a pollen tube grows, it grows from the stigma. Pollen tubes in angiosperms, much, much longer and faster growing than pollen tubes in gymnosperms. Okay. So, flowers. Since I mentioned the stigma and the ovary, flowers are a big deal. And that comes right into critters carrying the pollen. Why would critters carry the pollen? Because they go to flowers. And why do critters go to flowers? Because flowers produce nectar. Why do flowers produce nectar? So the critters crawl into the flower to consume the nectar. And what happens there? Well, we also have our little pollen-producing structures in the flower. So when the critter crawls into the flower to eat that tasty, sugary nectar, it gets pollen all over it, so it gets thoroughly covered in sperm. And then it can go off to another flower, and when it crawls in there and gets covered in more sperm, it delivers some of that sperm to the stigma. So, there we go. Definitely a stigma when it comes to reproduction in angiosperms. I'm so funny. I'm tired. Okay. Other things that are a preview of coming events that I'm not going to fully talk about right now. Endosperm tissue and a double seed wall. But we're literally going to get into this stuff. Angiosperms. 1. We refer to angiosperms as flowering plants. Hence, one of those shared derived features, one of those synapomorphies of angiosperms being flowers. 2. Ovaries. Right? Diploid tissue containing all them ovules, one or more ovules. By the way, you may notice in this diagram the ovary is down at the base, that sort of widened base of the flower where the flower meets stem. And if you've ever, looked at the seeds of a flower, like if you've ever had a flower growing on your porch and it actually seeded, the seeds are down here at the base. I wonder why. Sunflowers are special. They're a very shallow flower. They still have, seeds being cranked out near that base, but it's all sort of very shallow. I'm not here to define all of the weird exceptions or why they're not exceptions. Okay, so diploid tissue making up ovaries that fully enclose the ovules. So let's talk about the anatomy of a flower. All right. So let's talk about the female reproductive structures of a flower. So we'll ignore our whole combined thing there and just draw that. Okay. So this whole thing is a carpel. Fun times, though, it's also referred to as a pistil. So, good times. First off, we have the actual site of pollination, the stigma. Then we have that long, thin column connecting the stigma to the ovary, called the style. So, stigma to style, and then we have the ovary, and then inside the ovary we have, this drawing simplifies it by doing a single ovule, so why not? That is a terrible drawing. Whatever. The ovule. inside the ovary. Right? With the sporangium that has the sporocytes that produce the spores that produce the gametophyte that produces the egg. Okay. So, how does pollen get from flower to flower? Okay, so much simpler on the male reproductive structures, by the way. Much simpler, right? We have a long stem-like structure called a filament. And then we have the pollen-producing structure called the anther. Make the pollen. Pollen production happens in the anther. And the whole thing is called a stamen. So, in some angiosperms, it's wind. In most angiosperms, it's critters. How does pollen get to the stigma? Mechanical transfer. Something else moves it. This can be compared to the self-motile flagellated sperm in previous plants. I know I didn't really talk about it. I thought I had more slides on it, but I think I cut down gymnosperms for time. But cool things, cycads, they have flagellated sperm. So that's kind of wild. So some of these gymnosperms actually have flagellated sperm. And it sort of blends the line between seedless vascular and gymnosperms, really driving home the fact that gymnosperms were the first of the seed plants to evolve. So that's kind of cool. By the way, pine needles are still leaves. They are still leaves. And they are still megaphylls. They are highly reduced, but they're still full-on vascular leaves. So that's kind of neat. Okay. Brief aside. Okay, I'm over here. All right. Not too terrible. All right, so flat, like mechanical transfer of pollen to stigma. Transfer of pollen to stigma is certainly angiosperm specific, which we mentioned because we're talking about flowers. But mechanical transfer, something moves it rather than it moves itself, is seen in a lot of gymnosperms, specifically the ones that don't have flagellated sperm. So, Hence why it wasn't under the list of like synapomorphics. So anyway, pollen is mechanically transferred. Things that help prevent self-fertilization here, oftentimes the ovule is not fertile when the pollen is being actively produced. So if you brush your own pollen against your own stigma, it won't be able to fertilize it. A lot of times it's vice versa. When the ovule is fertile, when the egg is ready to be fertilized, the flower is not actively producing pollen. So a pollinator that arrives will only get pollen from another plant on there. So there are mechanisms to prevent self-fertilization in a lot of flowers, and a lot of it has to do with timing. Gymnosperms too, by the way. It's not just that ovulate cones are higher in the tree. Sometimes those staminate cones are ready to rock and roll before the ovulate cones are, and vice versa. So, kind of cool. Lots of mechanisms against self-fertilization. Not that it can't happen, but there are mechanisms that reduce its frequency enough that sex is like, sex between unrelated individuals is generally how we're getting new plants. Okay, let's talk fruit. Fruit is an ovary. Good times. Who doesn't love biting into a fresh, delicious ovary? I know that when I'm feeling really hungry, just grabbing an ovary and just biting into it, getting that burst of delicious fluid all over my face as I chow down on an ovary. Very satisfying. Fresh flavors. Okay, so fruits are ovaries. We know this because fruits contain the seeds. And the seeds develop from the ovule. So, if it's completely enclosing an ovule, it's an ovary. A fruit completely encloses one or more seeds, so it's an ovary. You would be surprised at how many things are ovaries. Here's a dog just covered in ovaries, having a bad time. Here is a mouse collecting delicious, delicious ovaries. Here's the angiosperm version of helicopter seeds, which are ovaries. Relatively, thin ovaries, but ovaries all the same. So, peach, ovary. Orange, ovary. Tomato, you better believe it's an ovary. Hazelnut, ovary, because seed. Kind of fun. Lots of things, lots more things than you probably thought get classified as fruit. So that's kind of cool. Yeah, the milkweed ovary splits open once the seeds are mature, and then the seeds are dispersed by wind. So not all fruits are to be eaten. Right? Some fruits have adaptations for being moved around by critters, just stuck to the critters. Some fruits have adaptations to protect the seeds until the seeds are developed, at which point they split open, right? Some fruits, a lot of fruits, have adaptations to travel through the digestive tract. And then here's some fruits that are directly adapted to wind transfer. Whereas here, so I am pointing, so these fruits are fruits that are adapted to wind transfer. This whole thing is a fruit. Whereas here, the pod is the fruit, and our little wind transfer things are seeds. In other words, there's a crap load of diversity in the world of fruit. A lot of fruit is to attract animals that will eat the fruit, and then in turn eat the seeds, and then carry the seeds far and wide, and then poop the seeds out so that the seeds are in their own delightful fertilizer. Okay. Angiosperms and gymnosperms are both seed plants, but there are some fundamental differences in their seeds. We've already talked about how ovaries completely enclose the ovule. Let's talk about some unique features of the ovule itself in angiosperms. Okay, first off, let's draw. Easy one to draw. Right? So, here is our ovary. Just somewhere outside. Right? And then here is going to be our ovule. And this is our double layer thick integument. So, double integument, just to think of it. Right? If one layer of integument gives you a seed that can go dormant for a very long time and be very well protected, well, double layers of integument mean even better. Right? Right? Has to. But no, that is a unique feature of angiosperms. It's an apomorphy of angiosperms is the double layer seed coat. So they're going to have a double layer of integument. Right? Another neat feature. Okay, so we have the integument. We have the gecko trying to go into the lumbar thing again. Bad gecko. Boop, boop, boop. Okay, let's do our fun roundup of tissues. Integument. Sporangium. Right? I should make this smaller here. We'll do this, and we'll have this right here. Right, okay, so spore wall. Right, there's a lot of sporangium because we produced 4 spores, of which 3 died back. Spore wall. And then orange is the spore. Megaspore. Right, so it's worth noting that the spore wall, the megasporangium, and that double integument are all destined to become the future seed wall. Right? Just a reminder. Okay. Now, that megaspore is kind of neat because we're going to have our egg in there. Let's go purple. And we're going to have our gametophyte, blue. And interestingly enough, this tissue surrounding the egg, the spore and gametophyte tissue, is what we call binucleate tissue. So it's this mix here, the tissue surrounding the egg, right? So let's hit this real quick. Okay. So let's just ignore everything else but the egg and the tissue surrounding the egg. Which is the nutrient tissue. Right? Basically, the mix of sporophyte and gametophyte that's between the spore wall and the egg. One cell thick spore wall. So nutrient tissue. Here, haploid. Right? So, not too bad. Haploid egg. All right, angiosperms. I'm going to draw this a little differently. So here's my egg nucleus. I'm going to point out my egg nucleus. Haploid contained in my egg. I'm just getting a little more specific. It's the same thing, but I'm emphasizing something else. And then we'll do purple because it's different. Here's one cell that's representing all the cells, right? Because this is all this nutrient tissue is cellular tissue, right? So, nutrient tissue. Right? And then, of course, the spore wall, which is part of that structure that will become the seed coat. But we're trying to be consistent here. Spore wall. So this mix of tissue where we had spore tissue and mostly gametophyte tissue, right? Here, it's binucleate. One cell, two nuclei. Right? So it's 2 in, diploid-ish. There's 2 nuclei in it. So, why am I pointing this out? Let's throw a pollen tube up here. Here's my gymnosperm pollen. One pollen nucleus, useless. Other pollen nucleus fertilizes. Right? So, 2 nuclei, but only 1 is used. All right. So, we'll have our angiosperm pollen. That is too small. Two nuclei, right? You get a, this is up at the stigma. And you're going to get a pollen tube growing all the way down, all the way around, all the way into here. And a fun thing is going to happen. Both sperm nuclei through the tube. One sperm nuclei fertilizes the egg. One sperm to egg. The other sperm goes to the nutrient tissue. One sperm to nutrient tissue. There are 2 fertilization events. In one fertilization event, it's what you think of when you say fertilization, sperm plus egg. The other fertilization event is one sperm to the binucleate tissue, and we get, what do you know, trinucleate tissue. So let's call this future nutrient tissue. And now we have fertilized 3 in endosperm. So the nutrient tissue in angiosperms has its own name. It's endosperm. And the big deal about fertilization in angiosperms is that it's so nice it happens twice. One sperm nuclei fuses with the egg nucleus and you get your diploid zygote. The other sperm nuclei fuses with our binucleate tissue and we get 3 in or triploid endosperm. So this means when we talk about angiosperms, some of the big features of them are double fertilization and the presence of triploid endosperm in the seed. Double fertilization and the presence of triploid endosperm in the seed. So, big seed differences. Double layer of integument, right? Binucleate nutrient tissue that gets fertilized to become triploid endosperm. Endosperm is actually significantly more nutrient rich than the haploid nutrient tissue in gymnosperms. So, angiosperm seeds can remain dormant for longer because they have higher quality food supplies. So they have better food, so the seed can remain nourished for longer. Essentially, they can digest less of their nutrient tissue at a time. So, pretty cool. So angiosperms are the world champions in dormancy. That's pretty cool. Okay, so there we go. That was that was from this slide. Double fertilization, because one sperm nucleus fertilizes the egg, one sperm nucleus fertilizes the nutrient tissue to create triploid endosperm. Angiosperms are the most diverse and widespread phylum of terrestrial plants. So, literally the most diverse and widespread. This is for a number of reasons. One, their seeds are more hardy, right? They have the hardiest seeds of all. So they can be distributed furthest and widest seed-wise, and the seeds can last in dormancy longest, right? So their seeds can handle even worse conditions. So that's a big deal, right? That double integument layer, producing a thicker seed coat. That endosperm producing better nutrients, right? Another one, flowers. Flowers allow much wider scale pollination, right? Wind knocking gymnosperm pollen out of the cones and into the air will only take you so far, right? A tree is going to maybe be able to fertilize the trees around it in a radius of tens to like a couple 100 feet if the wind's particularly strong, right? Very local fertilization going on when you rely upon wind. Pollinators can travel large distances, right? Especially when you get to the point where you have bird pollinators. There are flowers that attract birds, hummingbirds for one, and those can travel very large different distances. So your ability to attract pollinators via flowers, man, that amps up the distance between mates, right? Fruits, right? Those amp up protection of the embryo, and in a lot of cases have adaptations to aid in dispersal, whether it's stickers sticking to things, fun little flowing structures that help it float on the wind, or in a lot of cases, something tasty so that animals eat it and disperse. So they've got a really increased dispersal radius. They can disperse further. So they can reproduce far and wide from their pollinators. They can disperse far and wide from their fruit. And then they have that endosperm, as I mentioned, for that increased nutrient to really give your embryos a good start. A seedling gymnosperm and a seedling angiosperm are going to have differences in, let's just say, robustness. Okay. Next life cycle, which is going to appear on an exam for. Okay, this is the angiosperm life cycle. And again, we have sporophytes. We have sporangium and spores, right? We have, this is getting into how we get our egg and such, but basically we have an even number of cells in here, and I don't want to go into polar nuclei, but this whole thing about how our nutrient tissue has some weird 2 nuclei in there, that's our binucleate tissue. Anyway, so you can see 2 layers of integument. There's our egg. I'm trying to go basic here. Megaspore, egg, right? Then you have pollen and sperm, and the sperm fertilizes the egg, and we get, our seed and our embryo, and the embryo grows a new sporophyte, and there you go. And then we just need to add our specific stuff in here. Right here, we have the pistil with the ovary completely enclosing the ovule, the filament, or not the filament, the brain fart, complete and total. The style, ovary, completely enclosing ovule. Style, connecting ovary to stigma, site of fertilization. So there's our female structure in the flower, so we can focus over here. The ovule. The ovule is where we have integument that produces the megasporangium, that has the megasporocyte, which is the same as megaspore mother cell, interchangeable term. And the megaspore mother cell, the megasporocyte, produces the megaspore. Only 1 megaspore survives because it's made by meiosis, and meiosis produces 4 cells. 3 die off, 1 survives. Thunderdome again. And then that spore germinates, and we get our gametophyte tissue, which produces an egg and some other fun stuff. So this here, that orange, is the spore wall. So then we have the inside tissue, where we have our 2 polar nuclei. Our 2 nuclei are binucleate tissue. Let's not add more terminology. Our binucleate tissue and our egg. Right. Go back here to our male structure, which is simply a filament and an anther. Here's the anther, which produces the microsporangia, which contains the microspore mother cells, the microsporocytes, interchangeable term. Right? The microsporocyte undergoes meiosis to produce 4 microspores. Each microspore producing pretty much the same thing you're looking at here, right? The microspore, the microgametophyte tissue, and the 2 sperm nuclei. Only in angiosperms, both those suckers get used. So, angiosperms have 2 functional sperm nuclei. So here's our pollen with the sperm nuclei. Pollen lands on the stigma, which makes sense because that's the site of pollination. It grows a big old long pollen tube. The pollen tube has to go all the way from the stigma down the style into the ovary, sort of along the little ovary wall and then to the ovule, at which point, once it connects into the ovule and makes it through to that tissue, we get 2 sperm nuclei going in there, and 1 fertilizes the binucleate tissue, and 1 fertilizes the egg, and we have triploid endosperm and our first cell of the newest sporophyte generation, the zygote. So 2 sperm, 2 fertilization. And then we have our embryo contained within our seed. So there's our seed coat, there's our embryo. Fun anatomy in these little plants, right? So we have the buds, basically the terminal. So when you have that embryo there, if you remember your plant terminology, we talked about vascular plants having the apical meristem, roots and shoots, the apical meristem can be thought of as producing the terminal bud. So here's the apical meristem for the shoot, and here's the apical meristem for the root. And then an embryonic plant has two leaves that it will come out with. Every seedling always comes out with 2 leaves. The seed leaves, which of course this is science. They have to have their own term, the cotyledons. So that's pretty cool. I think I'm getting into plant anatomy stuff that I believe I have in the next lecture. This is just all about plant evolution. Okay, so let's talk about synapomorphies. So here's our outgroup. The ancestral state, the charophytes, chlorophytes. Here's our most primitive possible ancestor to live terrestrial plants. So this is our terrestrial green plants. Right, so chlorophyll alpha and beta. Right, then we have the cuticle and the plasmodesmata. Plasmodesmata occurring. Screw it. Let's just ignore this stuff, because what I'm mostly interested in are the synapomorphies that separate out our bryophytes, our non-vascular plants, from our seedless vascular plants, from our seed plants. Okay, so characteristics, the synapomorphies that produce our bryophytes. Cuticle is the main one we talked about. Antheridia and Archegonia are just products of alternation of generations. So, alt of generations we could put in here. We mentioned stomata as well. Okay, so those are a development here in the bryophytes. We're really not breaking synapomorphies out amongst the different bryophytes. So really cuticle, stomata, and this is the onset of alternation of generations, right? Seedless vascular tissue, or seedless vascular plants. Vascular tissue. Sporophyte dominant. The presence of vascular tissue in the sporophyte, right? So there we go. We'll ignore euphylls. That's a fancy term for true leaves, megaphylls. So we'll just ignore that for the moment. But we did talk microphyll versus megaphyll earlier on, whereas microphyll is thought to be a single stem, right? So you have the vascular tissue inside there, and it's like a stem that got widened out, whereas megaphylls were when you had, branches of a stem that got connected together with tissue. So one stem, multiple stems and branches. That's microphyll, megaphyll. So we could ignore euphyll, but just a reminder of the term that we did use, microphyll, megaphyll, and what they were about. I'm not inserting them onto this. Okay, seed plants. Amazingly enough, the seed. Angiosperms, flowers, fruits, and then, of course, some of those other things like double fertilization and the double integument wall and the ovary, right? Gymnosperms have ovulate cones and staminate cones. They have ovules that are exposed to the outside. There's lots of stuff you can do to fill in this sort of thing. And a lot of those synapomorphies are great test questions, just saying. Okay. What is it today? 5:40? Oh, I love you. Good night.